Follow us on social media and please hit the subscribe button so you don't miss out on any updates from our ALS experts.
All right, folks, welcome. It's another community gathering of your community. And each one of you in that little square is more important than you have any idea. So I just want to say thank you for your time, for your heart, and for everything that you're bringing here. Every little piece of the puzzle is very important. And each of one of you are a piece of the puzzle. So welcome. And with that, it's all yours, Lisa.
Thanks, McFinn. Welcome everyone to tonight's ALS talk. I'm Lisa Deegan, and I am part of the Everything ALS team. Our whole team has been affected by ALS. So for anyone joining this ALS talk for the first time, I just want to share why Everything ALS exists. It's really to pay tribute to those affected by this disease and the loved ones we've lost. So we're working hard so others don't have to suffer the way we did. And we're working hard to change the paradigm in ALS, working towards a cure by focusing on early diagnosis and progression tracking and advanced therapeutics. So after today's FDA meeting regarding Amylyx, I don't know if you guys saw that today, but it definitely shows that we should have some regulatory flexibility, and that our subjective instrument for measuring progression and drug effectiveness definitely needs some improvement. So this is exactly why Everything ALS is doing our research on digital biomarker discovery that can help make the results to be more meaningful and reflective of people's lives. So we can really use your help. Please sign up for our citizen-led research at everythingals.org/research. So I want to just thank you again for joining us tonight. We like to say that we are bringing the experts right to your living room, and we are bringing you guys directly, our ALS community directly to the experts. And we offer a safe place for everybody to have open discussions and be around others and meet others going through a similar journey. So speaking of the experts, top experts, we are so honored to have our guest speaker tonight, Dr. Robert H. Brown, back on our platform for the second time. And tonight he's gonna present on Antisense oligonucleotide therapy for ALS. Did I get that right, Dr. Brown?
You did indeed. You did it very well. Thank you.
Okay, oligonucleotide. Okay, so Dr. Brown is affiliated with Massachusetts Medical School and University of Massachusetts Memorial Medical Center, and was previously with Massachusetts General Hospital. He is an internationally known researcher and physician and has led the quest to cure neurodegenerative and neuromuscular diseases like ALS for many, many years. Renowned for groundbreaking research on inherited and genetic underpinnings of neurodegenerative and neuromuscular diseases, Dr. Brown has recorded significant discoveries in identifying gene deficits that elucidate how ALS causes neurons to die. In 1993, a team of researchers led by Dr. Brown discovered the first gene linked to the inherited form of ALS called SOD1. Thank you so much for being with us tonight. Please join me in welcoming Dr. Robert Brown.
Thank you so much. I really appreciate this chance to meet with everyone. What a wonderful community. What a wonderful sense of joint enterprise in solving this terrible problem. This is fantastic to be able to spend some time with you. Can you all see my full screen now?
Yes, we can.
Terrific. So I'm going to talk tonight about some of the newer approaches to suppressing toxic genes in ALS with a major focus on a particular kind of reagent called an antisense oligonucleotide, also called ASO. And I will mention a couple of other modalities beyond that. I think everyone on this call knows that there are substantial challenges in the way one has to think about ALS. It's clear that although the final clinical pictures are dismayingly all alike, with a rapid progression and the lack of therapy, there is clearly disease heterogeneity. We know that because there's phenomenal heterogeneity in the triggers of the disease and the multiple genes that can cause the disease. There are only perhaps 10% of cases which are inherited, but among that 10%, there may be 50 or 60 different gene defects that can trigger the same disease. It's a challenge as well because we don't make the diagnosis until fairly late on in the illness. That is to say, a year goes by between the first symptoms and the actual diagnosis in most centers in the United States and Western Europe. It's a challenge because it's hard to deliver drugs inside the blood-brain barrier, the iron curtain. Finding the right dose is a problem. As we all know, we need validated biomarkers that will help us without actually doing spinal taps, know where the disease is. And it's also true that conventional therapies, what we call small molecules, have significant limitations. They may be very good, for example, at inhibiting enzymes or stopping inflammatory processes out in, say, the periphery. But if one wants to modulate the way a gene is behaving, those small molecules have problems. So over the years, many of us have started with wonderful work with Tipu Sadiq way back in the early 80s, began to look at the question of whether or not there's an interaction between genes and environment that could trigger the disease, and whether or not that in turn will provide a roadmap with targets for therapy. And so we've been pursuing that, many of us, since the early 1980s. And what we now know is, as I've said, that there are several genes that can trigger the disease. And we know as well that they trigger a set of events like falling dominoes, so that one gene with maybe one nucleic acid wrong out of, get this, 3 billion, one being wrong can trigger the disease. And then downstream, in so-called secondary phenomena, there are series of events that converge that kill, unfortunately, motor neurons. Now, one of the reasons we all got into this way back in the early 80s was the hope that by understanding familial ALS, we would learn about sporadic ALS. And I can tell you that we're now realizing some of that vision. There is clearly an overlap between the two. Some of the important lessons from familial ALS clearly affect individuals with sporadic forms of the disease. We've also learned, as I've tried to show in the bottom of this image, that there are non-motor neuron problems, such as inflammation within the spinal cord, that participate in the process of the disease. So how do we understand this? Well, one of the points to make in my, there we go, I am now finally advancing, is that the downstream processes are incredibly complicated. You can take your favorite part of a motor neuron and study it, and it will be abnormal in this disease. And I'm not going to go through all of the points that are listed on this particular image, but I will tell you that there is enormous spatial and temporal diversity in the way this phenomenally polarized cell, the motor neuron, gets sick. So early on, mitochondria are abnormal out in the periphery of the cell body. Somewhat later, the distal axon, which is 3 feet away, starts to unplug. And then there's a host of problems that occur in the intervening places at different parts in the cell at different points in time. So this is very, very complicated. One of the things that has helped in recent years in deconstructing this are these stem cells we can get from skin or blood that allow one to make motor neurons in a petri dish. And that means that I can see a patient in the clinic in the morning, and in the afternoon go to the lab and study that patient's motor neurons in a petri dish. I find this almost like science fiction come true. But here's the point. We now do think we have a series of targets for therapy. And if we go way downstream to that very complicated map I just showed you, there are multiple targets for therapy that one could think about. But on the other hand, our hope has been that if we go way upstream and correct or somehow suppress the toxicity of a given causative gene, that might be beneficial. And that's really what I'm going to talk about over the next 15 or 20 minutes, the upstream gene therapies. And that brings us to a question, just of reviewing, if you will, what you might call the central canon or dogma of molecular biology, of course, and that is that in the nucleus of every cell in the body, there's a full copy of all the nucleic acids that make up the genome, the DNA. And the way that works, it stays in the nucleus, but it makes a template of itself in the form of RNA. And the RNA can act not only in the nucleus, but outside the nucleus in the surrounding cell, the cytoplasm. And there, among many other things, it can make proteins, which are one of the final workhorses of any cell and cell biology. So what happens if a DNA for a gene that is related to ALS gets mutated? What can we possibly do about it? How can we help? So let me just comment on that. So if, for example, a gene is missing, one can think about ways to replace the gene. And that's not so far-fetched. So there are studies now using a virus that can sneak its way, if you will, into the central nervous system, and once in, express a cargo, express a missing gene. So the dean here at UMass is treating patients with little kids, babies even, with Tay-Sachs disease, who lack a gene by putting the gene back. And will it work? We don't know, but it's very hopeful. But in terms of ALS, one of the problems is not that something is missing, but the genes are mutated in a way that makes them poisonous or toxic. So the question is, how can we correct that? At the level of the DNA, again, you see that here, perhaps you can see my cursor. One of the things that's very exciting, but not yet ready for clinical prime time, is editing the defective gene to correct it. And by gosh, if one could do that, one would have essentially a permanent correction that would solve the problem. But we can't do that yet clinically. But what we can do is employ some new methods to change the template, to change the RNA, so that it will not be there to make a sick protein to kill the motor neuron. We want to suppress, in the studies I'm going to show you this evening, the mutant RNAs. And there are several ways now to do that, which are getting ready for, and indeed are coming into clinical practice. And I can't go through them all, but I would just say that one of them is to use a virus, once again, to deliver to the nervous system a particular structure called a microRNA, which can then shut down genes. The other process that's being much more widely used is to deliver the so-called antisense oligonucleotides without a virus, just by themselves. They're just structures of maybe 20, 22 nucleic acids in a small bead or small chain that goes into the spinal fluid, gets into the brain and spinal cord, and can turn off genes. And then, of course, if there's a sick protein, and this is not the topic for tonight's discussion, but in theory, one could try to deplete that protein by using sort of molecular sponges, using antibodies that could bind and clear the protein. So just one word about ASOs. This is a picture of how they work. So what you see in black here is a gene which is making an RNA. And what you see in orange is an ASO, which is made to target one piece of the RNA. And what it does, in fact, is attract to it an enzyme called RNase H, which then chews up the RNA right there, and thereby prevents a full RNA getting into the cell to make a full protein. And one of the neat things about ASOs is they can be delivered, as I think I mentioned, into the spinal fluid very easily, much more easily than viruses, and they can permeate not only the outside of the cell, the cytoplasm, but also they get into the nucleus. And so they're very versatile, and that's one of the reasons they've become really very attractive in the search for therapies. So I'll just walk through 3 or 4 examples of current efforts at gene suppression in ALS, and then I'll stop and hopefully we can discuss this and other aspects of ALS therapeutics. It's an exciting time for us all. So I'm going to just quickly share some work we've done on the gene called C9orf72. And this will, in the end, get back to a discussion I heard before this started about Biogen's cessation of its own C9orf72 program. Hopefully we can discuss that. But here, the lesion is in this gene, funny name, C9, the 9th chromosome, ORF, open reading frame 72, that is depicted here, where you see a long line and little blue vertical bits, which are exons that are the working part of the final gene. And then on black, you see these so-called introns. And the key thing here, as shown in the upper left, is that embedded in this intronic part on the left are 6 nucleotide so-called repeats made up of 4 Gs and 2 Cs. And normally, all of us, if we think about the public as a whole, will have a few of these repeats, 6 of them, 10 of them, 12 of them, something like that. But for individuals with the mutations in C9 that cause ALS, there are hundreds of these repeats or thousands. I mean, we have some patients we see who have 3,000 or 4,000 repeats. And it's very easy, just from this very simple picture, to envision many ways in which these repeats could destroy the function of the C9 gene. I'll just call it C9 for short here. So this has been one of the things that we've been focusing on. And it turns out, just to get down a little bit into the granular details, that one C9 gene makes not one RNA, but 3 RNAs. And 2 of the 3, which I've shown at the bottom, labeled in V1 and V3, actually contain the expansion, but the other one doesn't. So the nice thing here is that if one can target these isoforms that suppress these 2 transcripts, as we call them, then in theory, one might ablate the toxicity. That is to say, if one can suppress these 2, that'll leave the healthy one remaining. And I would just emphasize that many groups have done this. Biogen has worked on this. Wave Life Sciences has done an incredible job with a novel kind of ASOs to do this in a special way. And all have shown pretty much in a petri dish, and some of us now in animals and in people, that in fact, this can be achieved. And the result is to turn off the C9 gene. So we put about 3 or 4 years into working on the ASOs that we've studied. We showed they work wonderfully in cells from humans and in mouse models of C9 ALS. And we showed also that they're not toxic in sheep or monkeys. And that prompted us to get into a human trial. And the one patient I will just mention to you here is a 60-year-old individual who has about 2,500 of these repeats. And as you might guess, a very positive family history. And I'll just show you the take home here, which is depicted in this slightly confusing slide. But the key point is that if you look at that red line, it shows the level of activity of the gene over time. And what you see in the blue arrows at the bottom are increasing doses of our particular C9 ASO, the result of which over time in this patient was suppression of activity of the gene by about 80%, which says to me, and I have other reasons to confirm this, that these genes can indeed be turned off in a person, in a human. Will that make the disease better? And I'll just tell you that in the one patient we treated, we saw hints of clinical benefit, but he was a very, very early patient. And so may not be applicable to the broad set of patients. And again, as we heard mentioned beforehand, only this week, Biogen released a press release saying that they are going to disband, they're going to discontinue their ASO for C9 therapy because they didn't see appropriate clinical benefit. And we don't know the details yet, but whether or not that will be a general disappointment across the field, or whether there's something special about different ASOs that might make some more effective than others, or whether the timing of treatment makes a difference. All of these things that will need to be sorted over time. Let me mention briefly another study which has come out recently, which is a beautiful piece of work by Neil Schneider at Columbia. He's been working with a family of patients that have mutations in a gene called FUS. We reported those back in, I think, 2008, 2009. They're mutations that cause standard ALS, if I can use that term, in adults, but aggressive ALS, depending on the mutation, in children even. It's unthinkable. But we've seen 14-year-old, 15-year-old, 8-year-old children who have mutations in this gene and get very aggressive ALS. So Neil's been wonderfully enough working on this and in conjunction with colleagues at Ionis, has been studying one ASO, which has the property of suppressing FUS in cell lines and in animals. And what you see here is the ALS rating scale over time in an individual who was treated with progressive doses, escalating doses of the anti-FUS ASO. And what you can see is that the disease did seem to stabilize somewhat with this therapy. The patient, unfortunately, went on to die of the disease, but what was seen at autopsy, to make a long story short, is there was very, very, very significant shutdown of the gene. Neil achieved what he wanted to achieve at the molecular level. And this was shown by a number of assays. I won't go through them all. I'll just show you one picture in the middle, which shows a normal individual or an individual who has the particular mutation and is not treated, as compared to an individual, this individual who was treated. And what you see is, admittedly, these are just a few high power views, but what you see is almost complete clearance of the offending protein. And that's confirmed by other methods called Western blots we could talk about later. So here's another example where there's clear suppression of the gene without necessarily a major corresponding clinical benefit. But I will tell you that Neil has now treated several patients, some earlier, some with slower disease. And there is a suggestion that there may be some very welcome clinical benefit from that as well. Let me say a few words before I stop about a gene we've all heard much about, and that's the SOD1 gene. This has been studied extensively, and like the others, it has a mechanism of poisoning the motor neuron, which we think is what we call a gain of function. It's not that it's missing, it's that it's there and doing something very adverse. So it too is a target for suppression as a possible therapy. And as we may have heard, there were some very exciting results 2 years ago that came out of a trial of an ASO for SOD1 called Tofersen, named after a wonderful clinician up here, Toby Ferguson, who helped develop it. Study done with Tim Miller and Merit Sokovich, in which in a small group of patients, they saw very good suppression of the gene. And you see here in the spinal fluid, it went down by about 36% in the patients at the higher doses. And importantly, in the most aggressive forms of the disease, which was really incredible, they saw much improvement in the ALS-FRS score, as I think the up arrow is meant to show here. And this came out in the New England Journal of Medicine, really a wonderful achievement. The issue is that in a follow-up study, which was reported this spring, the results were in the same direction, but clinically, shall I say, certainly not statistically significant and a little bit less impressive. They did indeed see some suppression of SOD1, particularly in the faster progressing patients, and particularly early on. That effect didn't seem to last as much as one would hope after the study was, the control study was over, and they went into the so-called open label phase. And in both the fast and slow progressing patients, they again saw suggestion of benefit in terms of improvement in the ALS rating score, but it was not as big as in the first study. And I'll mention, though I don't have a PowerPoint, that they looked at the biomarkers for this disease and saw a good effect in the biomarkers in terms of reduction of neurofilaments. So this is I think, a super important study, hugely to the credit of Biogen. Hard to know quite yet what to do in terms of administration, except potentially to find a way to use it earlier in the course of the disease. It clearly shuts down the gene. And I'm just going to conclude with another way of shutting down SOD1, which we've worked pretty hard on, and that's using a virus to deliver an equivalent reagent called a microRNA, basically to shut down SOD1. So we did a lot of work in developing this as a possible treatment. If you look at the bottom right, the green arrow here, sorry, the red arrow here, you'll see two brown boxes, which are the microRNA that target and hopefully turn off SOD1. We treated ALS mice going into both ventricles at birth and then followed them to see how they did. The results are kind of summarized here. Untreated mice live and rather completely die, all of them, by about 150 days. Treated mice live much, much longer. A treated mouse is shown at the bottom. It's not normal. You can see it's got a curve back, but it's almost 200 days old. And it's running around. It's obviously alive. It can rear up, as you see here, and it can climb upside down. So clearly, this is a benefit. And in this case, the suppression of SOD1 was about probably 60 or 70%, not even 100%, with a clear benefit. So we took this same ASO that we developed for the mice. We studied it extensively in normal monkeys and got very good suppression in monkeys. The distribution of the reagent is shown in red here, and the activity reduction is shown at the right. It's about 85%. And that convinced us to try this in a patient. And so the first case we treated, I'll mention quickly, was a 22-year-old who had a very aggressive form of ALS due to a particular so-called A4V mutation. We now call it A5V. Strong family history, rapidly progressive left-sided weakness. So we used essentially the same reagent. Here again, you see the microRNA in this group headed by Chris Mueller and myself clinically, also with Merit Zakovich, James Berry, and her wonderful ALS team here. And the bottom line is that we saw some clinical benefits. You see here, for example, over time, which is the x-axis, the strength, for example, for knee flexion and knee extension, as measured semi-quantitatively with the so-called ALS testing system. And what we were surprised to see was the kind of plateauing of the knee flexion on the previously unaffected side once it began to get weak, and actually an improvement in the extension on the unaffected side. The side originally affected, the left side, continued to weaken and did not respond. Again, suggesting we didn't get to it early enough. This is an interesting image of this patient who called me about 6 months after we treated him to say he'd recovered some function in his left foot. And even after that, he recovered some function in his left hand, which gave us, I would say, a very cautious optimism that this is a therapy worth pursuing. Now, what I want to show you, and then I'll wrap up, is that when we looked at his SOD1, as Neil Schneider saw, and as was the case with the C9 study, the gene was shut down. So this band here represents the amount of protein in the lower cord or the upper cord as compared to other patients with the same mutation or healthy controls who have much darker bands, meaning they have much more protein. So here again, the gene was clearly silenced, but the clinical effect, though there was strongly suggested, was not rescue of the disease. So that's where I'm going to stop. I think just by way of conclusion, it's stunning that the complexity of this disease is enormous. There are so many molecular events that participate in the evolution of ALS, starting way upstream, in some cases with single gene defects, and then a complex of pathologies downstream. We are learning a lot about sporadic ALS from familial ALS. It's clear from what I think, from what I've just shown you, that personalized therapies targeting specific mutations, specific genes, can certainly suppress the gene themselves. But we're not yet sure whether that will produce clinical efficacy, and it may well depend on timing, dosing, and other variables. The bigger picture, I think, is that these so-called biological therapies, using RNA, using viruses, using microRNA, using editing techniques, are, I think, going to play a very big role in our approach to this disease. One tiny example is the following. One therapy, which I've shown you, is to turn the gene off. In other cases, what's needed is to change the way the gene is put together, to change gene splicing, or even to increase the activity of a gene. All of these things will happen despite some of the downturn news over the last couple of days. I am very optimistic about where we are going. I think the face of this disease, the opportunities for new therapies has never been better and is going to make a huge difference as we go forward. So I took a little more time, I think, than I should have. I'll be very happy. I've got all night, got a beer here and some Ritz crackers. Spend the whole night. I can answer any questions you folks may have. So thank you for inviting me.
Thank you, Dr. Brown. My name is Sarah. I'm an Everything ALS team member. And we met the last time you were here, but I remember you just because I'm so inspired. So myself and our student ambassador, James, will be going through the questions with you. We have some in the chat as well, some that were sent to us previously. So just to get started, and you kind of touched on it already, but in the light of all the news this week, what are your thoughts on those trials and what may be next for the C9 cohorts of patients?
Yeah, so first of all, the good news in all of this is these genes are being shut down. Okay, 6 years ago, 4 years ago, we didn't know that could happen. So that's a very important lesson. Now the question is, I think, again, it's timing. When do we have to intervene? When is it too late to shut down a gene and see benefit? You know, one of the things that's underway in multiple centers, as I know you've all heard, is this effort, I think as a cluster, we're calling it Prevent ALS, to study people who carry the genes that we know with 95% certainty will cause ALS, and try to figure out what's the earliest manifestation. Will it be some digital marker that you guys are studying? Will it be a spinal fluid marker, a blood marker? And then to use that as the hint that we need to get in then and treat the patients. So I think I'm not discouraged in the sense that I think the good news is these genes are being shut down. Wave Life Sciences is doing it. Biogen has done it. The question for C9 is, when should we start? Okay. And the other point is, and it's a whole huge topic, obviously, but all of the things that go crazy, all of the chaos that occurs in different parts of the cell at different points in time, all of that has to be dealt with. The analogy, which is totally obvious, which I use a lot, is if you light a fire in a house and you burn the house down and you blow out the match, the house is still burning. Now there's everything else is still going on despite the original trigger. So we will clearly have to combine gene suppression, I think, with substantial downstream intervention. And the question is, what's the Achilles heel? What will we have to target downstream? One thing, two things, five things? We could go on and on. We've all, I think, in the last few years seen wonderful progress, for example, in some kinds of cancer by using polydrug therapy. You can treat lymphomas, you can treat childhood leukemias, which used to be uniformly lethal with combinations of drugs. And so far, there have not been many combination drug therapies in ALS. So let me pause there.
Dr. Brown, thank you for rejoining us again. A wonderful presentation. I think in light of the news this week with Biogen, it was nice to see some hopeful work being done and accomplished. So I'm going to move on to the next question, which ties into that, which is, what currently personally gives you the most hope for the future of ALS from what you are seeing?
Yeah, well, I have several lines of thought. So one is that these gene interventions in terms of suppression work, it's a whole nother story, but gene modulation, one of the themes that's emerging in ALS is that when some of the genes, like a thing called TDP43, become abnormal, a lot of other genes, probably hundreds of them, don't get put together properly. So if we can find out which of those are most important and use these therapies to change the splicing or the putting together of some of these other genes, I think that will be helpful. I spent most of the day listening to this FDA panel on Amylyx. So the thing about Amylyx, just briefly, is that this was the first really well-controlled multi-drug study in ALS, and it showed very promising results. We could go around at length on whether or not the FDA was right in its decision or the panel was right in its vote. I happen to think there ought to be a mechanism for provisional approval of drugs like that, so they get a chance to both benefit people and be studied further. But there's not a good pathway for that at the FDA. But here's the point. When you begin to combine drugs as they did, I think we'll see much more opportunity for benefit. The other thing that I'm sure everyone knows is that there are some new models for drug trials. So the folks at the MJ should put together this, parallel processing, shared placebo, 4 or 5 arm drug trial, which will speed everything up and be much more cost effective. So all of these things, I think, bode well.
Thank you. Our next question is kind of speaking to the people who are on this call that are looking for the next thing to potentially try and either join or see if they qualify for. Can you speak to the timeline of your study and what may be the next steps in the trials?
Yeah. Well, we are trying to digest the news that has come out this week. I mean, I don't know yet. I'm not privy to a lot of the details about how much, for example, the markers of activity of the C9 gene were influenced by the particular ASO used by Biogen. We see 80% reduction. Did they see 20%, 70, 80%? I don't know. So those kinds of questions need to be answered. We have, the interesting thing is, I have a protocol which is all written to expand our work with that particular ASO to a larger group of people, 20 or 30, which is still a very small study, but at least to get to the next level. And we're going to hold on that until we learn more about why the Biogen trial did not succeed, if we can get the information.
Thank you.
I know when it comes to genes, and as individuals, we're kind of at the mercy of luck of what genes we end up with in life. Environmental factors are a little bit different. So in your opinion, are there any or strongly possible environmental factors that may affect C9 or SOD1 that's leading to ALS? And if so, which ones do you think they may be?
Yeah, you know, I don't really feel I can speak confidently about that. I mean, you probably know the data better than I. There are some environmental factors that really are thought to be associated with higher risk of ALS, and even in a dose-dependent manner. Smoking, for example, 3 or 4 studies have shown that, curiously enough, that enhances the risk of ALS. But what I haven't seen, you ask a super question, is are there particular environmental factors that operate in the context of particular genes? Great question. And again, that's why groups like yours coming together and generating large numbers of folks who are willing to share the data of their life and create epidemiologic repositories will make a difference, because we just don't have the data to really tackle what is a super question.
The next question talks about oligonucleotides specifically, and is there any negative effects to turning off a gene like that?
Well, there can be, and so part of the answer is dependent on how you turn the gene off. For example, do you turn off both alleles, meaning the good gene and the bad gene, or do you target just the bad gene? So one of the neat things about C9 is there are 2 genes, one has a mutation, and the one that has a mutation still makes 1 good piece of RNA and 2 bad pieces. So if you just get those 2 bad pieces, even the gene with the mutation is still making a normal product, and the other gene is making 3 normal products. So again, a lot depends on whether you're specific for the mutation or not. If in general one shuts down both copies, there are senses in which that could be adverse. For example, it depends a lot, I think, on the age at which you treat patients. Just to come back to SOD1, there are now some reports that babies that lack SOD1 have very serious motor developmental abnormalities. Okay, it's not good to not have SOD1. An adult in which you shut down SOD1 by 50%, maybe that's okay. But we don't know, we don't have a qualitative or even, we don't really have a good answer to your question. So that remains to be seen.
Thank you.
If it is hypothesized that ALS is due to a gene mutation like C9 or SOD1, has there been any evidence of a sporadic ALS case due to a gene mutation becoming familial ALS and passed down?
Yes, there are some examples of that, to be sure, not many. We have a few SOD cases where we find clear mutations in an affected individual, and mom and dad don't have the mutation. But by and large, we don't see that. Okay, now, but to turn that question around a little bit, in the FUS cases that affect the little kids so aggressively, almost every one of those is a new mutation. In almost every one, mom and dad don't have the mutation, but somewhere in the sperm or the egg, the mutation arises and it leads to the disease. So that's my answer. Most of the time, we don't see, I would say, new mutations.
A couple of questions in the chat about the ATAXN2 study. I'm wondering if you could tell us a little bit about it and also tell us your thoughts.
Yeah, well, I think it's an important study to be done. The observation is made that this so-called CAG repeat, this structure polyglutamates in the gene can get slightly expanded and become toxic. If it gets really expanded, it causes a major cerebellar disease. But in the intermediate size of expansion, it seems to run a risk for enhancing ALS prospects, probably by, among other things, influencing things like this gene called DDP43 and what's called stress granules in the cell. So it's an important gene. And as I think you know, its importance is that it's relevant to sporadic ALS as well as familial ALS. And there are a couple other genes like that as well. And that's, again, what begins to give us hope, because looking through the keyhole of familial ALS, we're getting a sense of the broader universe, the broader space of how sporadic ALS at least is influenced. And genes like ATAXN2 have popped out of those kinds of studies. So I think that's a very important study.
We've spoken about C9, SOD1, and FUS. Is there any work being done on TAR-DBP familial gene as well?
There are people working on that, yes. And the question there is, in fact, there are many lines of approach, but the question there is, will this sort of suppression therapy that we just talked about be effective or not? And There are some data suggesting that reducing the TDP-43 by 40 or 50% is actually very adverse. Similarly, increasing it, overexpressing it, even wild type or normal TDP-43 is also very adverse. So the notion there is that one has to calibrate just what kinds of changes in level will be tolerated and potentially beneficial. There is some view that it, quote unquote, autoregulates itself. So the hope is that if you turn off the sick allele, maybe the good allele will make more and it'll come out balanced okay. And I think that view does justify thinking about targeted suppression of the mutant allele as one approach. But it's very early days. I don't know of anyone who's about to put that in patients, but I'd like to be very wrong.
Next question kind of stems off the last two. You spoke about how not having SOD1 in childhood is detrimental. But the question is, how then does that make sense for why it builds up in adults? I guess there's some confusion on the progression later in life.
Yeah, so just to be clear, the disease it's essentially in utero when babies don't have SOD1 is a complete loss of function disease, okay? Whereas the disease in adults that we associate with ALS is not a loss of function disease. There is still SOD1 doing the job of SOD1, but in addition, the mutant molecule has done something else which is poisonous. So, the actual mechanisms are probably very different. But the worry still is, and you asked the question, if you really shut down SOD a lot in an adult, is that a bad thing? In the fellow I showed you that we treated, if we hadn't shut his SOD down, we shut it down by 90%. If we'd only shut it down by 50% and only the mutation, would he have done better? And I think he might have. We're kind of trying to figure out how to take that forward. Good question.
Thank you.
On which of the ALS FRS scales did tofersen do well and not so well? For example, SVC, FVC, etc.
Yeah, I can't give you a good answer to that. I'd have to go back and look at the fine print. I think in the follow-up study, there was a hint and a trend with several of the markers, but they just didn't reach statistical significance. But I can't give you a very granular answer.
The next question is about your APIC bio treatment.
Yeah.
The question is the trial still proceeding, and what phase is it on, and any
So that is an extension of the very therapy that I just showed you in the one young man. So that's the concept behind the APIC trial, and I expect they can be doing the first case probably by the summer or the early fall.
Wow. Okay, that's great.
I know it seems like it's in its early stages from what I'm hearing. So I don't know if we'll be able to have an answer for this question, but are there any risks observed yet associated with the APIC treatment for SOD1 ALS?
Yeah, I think, the thing about these viral treatments is that they require a lot of virus to get a lot of the microRNA into the CNS, basically, into the motor neurons. And in fact, in our study with that young man, we used, I think the number is about 100,000 billion viral particles. That's a huge burden of particles. And what we found in that first case is that the challenge, the burden, the load of the virus triggered an immune response, which was problematic. And one could say several things. The first is that, of course, if it cured the ALS, we'd deal with it. But it turns out we can deal with it anyway. And we do that now by suppressing the immune system. So for example, in these babies with Tay-Sachs disease that are being treated here, we suppress the immune system. And what in the second case we did with the virus, we used what essentially you would use for a lung transplant or heart transplant, major immunosuppression, just for about 3 to 4 months. And that completely took away the neuroinflammatory adverse features. But yes, there are challenges. There are challenges with ASOs. I mean, ASOs can also provoke inflammation. There are reports of rather dire inflammatory and even so-called necrotic changes in some of the sensory ganglia. There are reports in some studies, although exactly how it occurs is unclear, of atrophy of the brain and slight enlargement of the ventricles. So I would say the last chapter is certainly not written about ASOs either, although by and large in the 3 or 4 studies I've just mentioned, they're pretty safe. But these are great questions.
The next question talks about if we know when DPRs are started in C9 versus TDP43 pathology. That was all that was written. So I don't know if that gives you enough information for the context.
So these so-called dipeptides, what I showed you that picture of this huge C9 hexanucleotide expansion. So one of the things that happens is that somehow gets cut out in the nucleus from the gene. And as a big RNA structure, it gets out of the nucleus and goes into the surrounding cytoplasm. And there it engages with a protein making machinery and it so-called ribosomes, and it makes protein. So it's a different protein than the normal gene makes, but it's a protein which is quite distinctive. It makes actually 5 different proteins, depending on which piece of RNA goes out, and those can be toxic. So that's a very important question. They also are a marker for how active the gene is. So we use that as our marker for gene activity. We think that the polydipeptides are probably made very early in one's life. I mean, I don't know that we have a true look at really young patients versus older patients, but it's clear that even asymptomatic carriers of C9 make the dipeptides. So then you can ask what triggers their being toxic, if they are toxic. But you can also ask in any one of these ALS gene mutations, why does the disease not start till age 50 or 60? Why does someone live a half a century without disease and then bang, the disease starts? Is that because over time there's more of the gene expressed? Is that because the defenses decline with aging, which they probably do? Is that because with aging, there's a general propensity for there to be more inflammation in the brain? That's true. Is it because with aging, there's a propensity for proteins to misfold? That's true too. And another thing is that cells that the subcellular organelles that produce energy, mitochondria, accumulate mutations with normal aging. So you've got a mutation which may itself not be more toxic, but all the adaptive processes may decline with aging. It sets the stage for the disease. I'm speculating, but that's the kind of scenario that I think we're dealing with.
This next question, I think, is applicable to a lot of trials we see, but it may be specifically for SOD1, so bear with me. Even if there isn't a big clinical benefit shown for far-gone diseases like ALS, why aren't patients able to have greater access to shut down the genes to slow further progress in SOD1 patients?
I'm not sure I understand the question. Is the question access to the trial?
It might be, or it seems like when there's a trial done that is deemed not statistically beneficial, even if some people do get benefits from it, why aren't others able to also try that out as well?
Yeah, I mean, these are super important questions about trials, aren't they? I mean, one of the things we know from the genetics alone is that there's this huge heterogeneity. So when you treat, 300 people with, say, sporadic ALS, or even until now, we've not really even checked patients in a large trial. We don't know if they all have the same disease. So that's one reason why some people may respond and others don't. There are many other possible reasons. So-called non-genetic or epigenetic factors may play a role. I think, there are, we have 3 billion nucleic acids in our genome, and roughly every 1,000 base pairs, there's a variant. And so there are millions of variants in DNA in all of us sitting here in this Zoom call. And who knows, maybe and this is likely, there are particular combinations of variants that make one either more resistant to or more susceptible to the benefits of a drug. So those are some factors. We already know retrospectively that there are some known modifiers. There's a thing called UNC13A, which you may have heard about, which modifies the speed of and even the risk of ALS. And it may be that modifies the degree to which some drugs will affect patients, such as lithium, early days. And that's a big criticism, no one's particular fault, because the tools haven't been there, but a big criticism of most of the studies we do is that we don't know the genetic profiles of patients. And therefore, it's very difficult to pick out whether or not there's a subgroup that may really respond versus others. Key issue.
I'm just learning so much. I'm like absorbing as fast as I can. The next question is, you talked about earlier about passing it down de novo mutations or new mutations to offspring. And the question is, how often does that actually happen?
Yeah, so as I say, it's pretty rare. In the FUS cases that I mentioned, it's 100%. So the little kids with super aggressive FUS, this is according to Neil Schneider, whom I deeply respect, those are all novel mutations. But for most of the adult onset cases, and certainly for SOD, that's very rare, yeah. Now, there's another possibility that does not get looked at very much, and we're interested in that is, what if the mutations occur in the spinal cord and not in the blood cells?
Yeah.
Okay? That's something to think about. But in general, we think that process is rare.
When you shut down the genes, I'm assuming it's SOD1 and C9 this individual is talking about, can you determine right now if the damaged neurons can heal and repair themselves?
Yeah, that's another yet another of the really key questions here, and we cannot really. Now, we don't have a good way to sort that question out, because again, there is a sense that the complexity and harshness and adverseness of these downstream events may eventually make a cell beyond rescue. And and we also know when you look at autopsies that different populations of motor neurons are dying at different rates. So even in one person at death, you can see some that look pretty good and others that are devastated. So this gradient of involvement is really key. And I don't think we have a good way to answer that question. The presumption is you get to people earlier, there are more to be rescued. And that's why you see most studies requiring that people have pretty good ability to breathe and potentially even the ability to walk, which is ultimately very, very unfair for later stage, long duration patients who are automatically then excluded from studies. But of course, the studies are done with the intent of showing the biggest effect in the shortest period of time. That's a key question.
The next question is, would you mind commenting on Dr. Bedlack's work with ALS reversals?
No, it's very, very important. I talk to him about that periodically. It hasn't yet revealed, as we hope it will eventually, what causes some people to be reversible. But there are some very bona fide cases. We need to study them more. Simple enough. I mean, for example, just give one example, we haven't, done big DNA studies. We haven't done biomarker studies. He's done the most of anyone to try to put them together and make sense of them. For that, he gets huge credit for that and many other things he does. But I think, the old biologists used to say, treasure your exceptions, because it's the exceptions that teach you the broad rules. And it is those cases that I think may ultimately, if we understand them, prove incredibly informative.
I agree. Moving on to the next question, what small molecules are in studies for C9 specifically?
Yeah, I don't want to comment on those now. There are some that modify sort of epigenetic characteristics of the gene and therefore potentially its expression levels, but I don't want to comment on that now.
Is TDP-43 suppressed in the C9 and FUS studies?
Yeah, that's a good question. Not to my knowledge. Not to my knowledge.
Interesting.
What's interesting is that there seems to be a bit of a hierarchy in the sense that the kind of missplicing that you see when TDP-43 goes awry can also be seen, but perhaps less pronouncedly so, when C9 goes awry, as if there's something about an interplay between C9 and TDP-43, such that TDP-43 does not work normally when C9 is mutant.
And this will be my last question before Sarah asks the last question. So thank you for joining. My last question is going to be, the Prime C is in trial. Could you speak to this as a promising trial for all types of ALS, or what are your general opinions and thoughts on it?
Yeah, so this is the combination of two. This is another dual, right? It's two essentially antibiotics. I think it's very exciting. I mean, there was the one study that Vivian Drury did in Israel, where she had essentially natural history controls and compared them to people treated, and she saw some benefit in terms of slowing of the progression. I think that's just hugely exciting and obviously has to be taken forward. I mean, how remarkable if two drugs that have been around for 30 years each in combination have a benefit that we've missed all these years. I think greatly to her credit and the colleagues she works with who discerned this, I think first in worms. I think this was small animal studies that disclosed the possible benefit of the two together, which tells you it seems to be a mechanism that works over multiple species, which is also quite exciting.
And then our final question, because it is 8 o'clock and we do want to respect your time, is you mentioned during your speech that you were waiting to find out a little bit about the Biogen's results before continuing your C9 study. Can you just give a little bit more clarification as to what that looks like?
Yeah, I mean, there's nothing terribly profound here, but I just love to see the profile of how well their molecule worked on suppressing the activity of the gene. You know, looking at, again, this dipeptide as an output. My guess is they probably had good suppression, because my further guess is they probably would not have proceeded if they didn't. But I would just like to know those details. Was there something peculiar about the age distribution or the timing of intervention? I mean, the one patient we got into happened to be in the very earliest stages of the disease, and it maybe that's again where we have to go to achieve some benefit. So those are the kinds of things that I think are interesting, and they're very obvious, but I think they're important. And as always, Biogen, I'm sure will do a great job in sharing the information and reporting it, which will really help the whole field. I mean, to their credit, they have a full study there. I mean, they can draw some firm conclusions about failure of clinical a response, and it'll be nice to see it all disclosed in the light of day, and I'm sure they will.
Absolutely. Well, Dr. Brown, we want to thank you for coming and giving us so much information. I know I will be rewatching this to be able to look at things up and read my notes as to what I learned, but we always learn so much when you come, so we're very grateful for your time and your expertise.
Well, thank you. It's truly my pleasure, and as I scan the Zoom frame, it's great to see so many friends and colleagues again, and great to be here. Thank you so much.